Hello, everyone, and thank you for being here. I'm Brad Evans, and I lead disease state scientific strategy at BioIVT. Today, I want to focus on something that has quietly become one of the biggest determinants of success or failure in liquid biopsy studies, and that is pre-analytical control. This talk is not about the newest sequencing chemistry, and it's not about AI classifiers or advanced bioinformatics pipelines. Those technologies are moving incredibly fast, but what we are seeing across the field is that analytical sensitivity has improved faster than our control over the samples those assays depend on. So today, we're going to focus on what happens before the sample ever reaches an analyzer, because increasingly that is where a signal is either preserved or permanently lost. And once it is lost, no downstream technology can bring it back. Let's start with why this has become such a critical issue right now. Here's what the talk's going to be going over. First, I'll explain why pre-analytical variables now determine assay performance more than the assay itself. That shift is subtle, but is happening across MRD, early detection, and multi-omic biomarker programs. Then we'll look at the four major analyte classes in liquid biopsy: circulating tumor DNA, ctDNA, cell-free RNA, including microRNA, extracellular vesicles, and proteins. And I'll walk through how each of these fails under suboptimal handling conditions, because they do not fail in the same way. From there, we'll translate that biology into practical decisions. Which two chemistries do what? How centrifugation changes plasma composition, why time and temperature are biologically active variables, not just logistics. We'll then compare single analyte workflows with multi-omic workflows, because those are fundamentally different design problems and require different playbooks. And finally, we'll talk about pre-analytical QC, the kind that prevents false negatives and misleading biology in clinical studies before expensive analytical testing ever begins. To understand this now and why it matters in this moment so much, we need to zoom out first. Over the past decade, analytical sensitivity has improved dramatically. MRD assays can now detect tumor-derived variants at extremely low allele fractions. Early detection tests are searching for tiny signals of disease in a vast background of normal DNA. But pre-analytical control has not improved at the same pace. We are now in a situation where the assay is more precise than the sample is being given, and that creates a scientific bottleneck. At ultra-low signal levels, background noise from leukocyte lysis, RNA degradation, vesicle release after collection, or protein modification during handling can exceed the biological signal we are trying to measure. The assay is working perfectly, but is faithfully measuring a distorted input. In multi-omic workflows, this compounds. Each analyte responds differently to handling conditions, so variability increases in ways that are hard to interpret later. What looks like biology can actually be a pre-analytical artifact. So increasingly, the limiting factor in liquid biopsy performance is not sequencing depth or algorithm performance, it is sample integrity. So where does unwanted variability actually enter the system? This slide is the foundation for everything that follows. It shows the full pre-analytical chain, every step where variability can be introduced before we ever go to sequencing, PCR, or any downstream assay. The key idea is that variability enters early, the effects accumulate, and once the sample reaches extraction and QC, it is often too late to fix what went wrong upstream. Let's walk left to right. It starts with the blood draw itself. Venipuncture technique, order of draw, and how the tube is initially mixed all matter. Poor technique can cause hemolysis or platelet activation, and that immediately changes the molecular content of the sample before it even leaves the clinic room. Next is tube chemistry. This is the first chemical environment that blood experiences: EDTA, cfDNA stabilizers, RNA stabilizers. Each of these actively modifies cellular and plasma biology. Some prevent leukocyte lysis, some inhibit enzymes, some fix cells. These are not passive containers. They define how the sample will evolve over time. Then we move into transport and delay, time to processing and temperature, room temperature versus cold. These determine how much leukocyte lysis, RNA degradation, and ex vivo vesicle release occur. Delay does not just reduce quality, it changes composition. Centrifugation is next. The force applied, how many spins are performed, and break settings determine how many residual cells and platelets remain. That affects genomic DNA contamination, platelet-derived vesicles, and protein carryover. After that comes plasma definition, whether a site uses single-spun plasma, platelet-poor plasma, or platelet-free plasma means they are working with different biological materials, even if they all label it plasma. This is a major but often under-recognized source of cross-site variability. Then we have storage. The difference between -20 and -80 degrees and how many freeze-thaw cycles a sample experiences can affect protein stability and vesicle integrity. Storage is not neutral. Finally, we reach extraction and QC. Here we measure cfDNA yield, fragment size, RNA metrics, hemolysis index. But the problem is that the QC is often applied too late. At this stage, we can detect problems, but we usually cannot reverse them. At the bottom of this slide is an important contrast. What is typically controlled in studies is sequencing depth, bioinformatic pipelines, and assay chemistry. What is often underspecified are tube choice rationale, time to processing limits, centrifugation parameters, plasma definition, and freeze-thaw history. So the message of this slide is simple. Pre-analytical variables are not background noise. They are design variables. If they are not defined and harmonized, variability is built into the data before analysis even begins. To show that this is not just conceptual, the next slide walks through real data examples of variability introduced at each of these stages. On the previous slide, I showed the conceptual pre-analytical chain. This slide shows real data demonstrating that each of those stages measurably changes what we end up calling biology. On the top left for stage 1 for the blood draw, this panel shows the effect of hemolysis. When red blood cells rupture, intracellular components spill into plasma. You can see clear signal differences between non-hemolyzed and hemolyzed samples. For RNA and microRNA, this is especially critical because many of those molecules are abundant in blood cells. So poor draw quality can create artificial biological signals that look disease-related but are actually collection artifacts. In the top middle for stage 2 for the tube chemistry, here you see cfDNA related measurements changing over time in standard EDA tubes versus stabilizing tubes. The key point is divergence over time. In EDTA tubes, leukocyte disruption contributes genomic DNA and shifts the signal. Stabilizing tubes extend the window, but they do so by chemically altering cell biology. Tube choice is not neutral. It defines how the sample evolves after collection. In the top right for stage 3, for transport and delay, this panel compares archived samples with those collected under a tightly controlled protocol. The controlled protocol shows a tighter distribution, while the archived samples show more spread and outlier. Delay does not just shift averages, it increases variability. For low signal applications like MRD, that added spread directly translates into reduced sensitivity and inconsistent longitudinal trends. On the bottom left for stage 4 centrifugation, different spin protocols lead to different levels of platelets and extracellular vesicles remaining in the plasma. Residual platelets can release vesicles after collection, inflating EV measurements artificially. More aggressive spins can reduce vesicle counts. So centrifugation does not just clean up the sample, it changes what is biologically present. In the bottom middle for stage 5, defining plasma, this diagram makes an important point. Plasma is operationally defined. The supernatant after one spin is not the same material as platelet-poor or platelet-free plasma after additional spins. These fractions differ in residual cells, platelets, vesicles, and protein complexes. If sites use different definitions of plasma, they are not analyzing the same biological compartment. In the bottom right, for stage 6, for storage, here you see the impact of freeze-thaw handling on vesicle concentration and size. Storage is not neutral. Freeze-thaw cycles can change vesicle characteristics and likely affect other analytes as well. How you freeze and thaw becomes part of the biology of the specimen. So across this slide, the message is simple. Every stage in the pre-analytical chain can produce measurable changes that are large enough to obscure or distort the biological signal, especially when that signal is rare. That is why these steps must be defined, controlled, and aligned with the analyte of interest. Now that we've seen how variability enters at multiple steps, let's zoom in on the first truly irreversible decision point, tube chemistry. Liquid biopsy is often discussed as if plasma is a single uniform material, but biologically that is not true. ctDNA is fragmented and present at very low abundance. Its main failure mode is contamination from genomic DNA released by white blood cells after collection, which dilutes tumor signal. cfRNA, including microRNA, is enzymatically fragile. RNases are everywhere. And additionally, cellular stress after the blood draw can release large amounts of intracellular RNA that confound interpretation. Extracellular vesicles are intact lipid-bound particles. They can be artificially released after collection by platelet or leukocyte activation, which can create vesicle populations that do not reflect in vivo biology. Proteins have their own vulnerabilities. They can be cleaved by proteases, lose post-translational modifications, or disassociate from complexes during handling. So when a protocol is described as optimized, the real question is optimized for which analyte, because they do not share the same stability rules. And the first place these pathways start to diverge is at the collection tube. Up to this point, we've talked about variability in the workflow. Now we arrive at the first truly irreversible decision point in liquid biopsy, and that is tube chemistry. The moment blood enters a collection tube, the biological environment changes. Tubes are not just passive containers. They actively and immediately modify cellular and plasma biology. Some chemistries allow cells to remain metabolically active for a period of time. Others chemically stabilize or fix cells to prevent breakdown. Some inhibit nucleases and proteases. Some introduce polymers or other additives that change the chemical composition of plasma. All of these interventions shift the balance between extracellular analytes and molecules released from cells after collection. These effects cannot be corrected downstream. If genomic DNA contaminates cfDNA because of leukocyte disruption, that signal dilution is baked in. If vesicles are altered by fixation chemistry, their surface biology is already changed. If proteins are chemically stabilized, their native interactions may not reflect the in vivo state. So tube chemistry does not just influence stability, it defines the biological starting material that your downstream assay will interrogate. That means multi-omic liquid biopsy cannot be treated as a single tube problem by default. It requires explicit analyte prioritization. You are deciding which biology to preserve and which trade-offs you are willing to accept. On the right side of the slide, you can see examples. Standard EDTA preserves a more native state but requires rapid processing. cfDNA stabilization tubes prevent leukocyte lysis but chemically modify the plasma environment. RNA tubes lyse cells and stabilize intracellular RNA, fundamentally changing the plasma compartment. Protein stabilization tubes inhibit degradation but alter enzymatic and molecular workflow. So the key takeaway here is that tube chemistry is a biological decision, not just a logistics one. Because each tube chemistry reshapes the sample differently, the next slide translates this biology into something very practical: how common tube types functionally align or conflict with different analyte classes. On the last slide, we talked about tube chemistry as the first irreversible decision point. This table translates that concept into practical guidance. Across the top, you see the major analyte classes: ctDNA, cfRNA or microRNA, EVs, and proteins. The check marks indicate relative compatibility, and the mechanism column explains why. Starting at the top, standard K2 EDTA tubes are actually quite versatile for short processing windows. They preserve native protein states and work reasonably well for ctDNA and vesicles if processed quickly, but they do not protect RNA over time. So EDTA is about speed and minimal chemical interference, not long-term stabilization. Next, the cfDNA stabilization tubes like Schreck, Roche, and Kaigen. They are designed to prevent leukocyte lysis and genomic DNA contamination, which is excellent for ctDNA. But they achieve this through cell stabilization chemistries that can alter protein biology and disrupt vesicle integrity. So these tubes are optimized for DNA, not true multiomics. If you move down to the RNA tubes like PAXgene, Strec RNA Complete, and Tempus, you'll see strong compatibility for RNA, but poor compatibility for plasma-based DNA, vesicles, or proteins. That's because these tubes lyse cells immediately and stabilize intracellular RNA, which fundamentally changes the plasma compartment. Then at the bottom are protein-focused tubes like BD-P100, Roche P100, and BD-P700 or P800. These contain protease and sometimes phosphatase inhibitors to preserve protein structure and phosphorylation states, but they are not designed for nucleic acid workflows. So the message here is simple. Tubes are not interchangeable. Each one is engineered to protect certain analytes by chemically changing the biology of the sample. That means tube choice must be aligned with your primary analyte, and if you want multi-omics, you often need parallel tubes rather than a single compromise. Now that we've defined what different tubes actually do to the sample, we can talk about how centrifugation further shapes what ends up in the plasma. Now that we've talked about tube chemistry, the next major determinants of what ends up in plasma is centrifugation, and the key idea here is simple but very important. Plasma is not a single uniform material. Plasma is defined by how you spin it. Let's start on the left with single-spun plasma. This is what many standard clinical labs do, one centrifugation step at moderate speed. The issue is that this leaves behind residual leukocytes and a high platelet burden. Those cells and platelets continue to release DNA, RNA, and vesicles after the spin, so the plasma composition continues to change over time. For ctDNA work, this means dilution of tumor signal with genomic DNA from leukocytes. For EV work, it means platelet-derived vesicles can dominate the signal. In the middle is double-spun plasma, often called platelet-poor plasma. Here you have an initial low-speed spin, careful transfer of plasma, and then a higher speed spin to further reduce cellular carryover. This improves ctDNA signal-to-noise and gives more consistent plasma composition across sites. It reduces, but does not completely eliminate platelet and cell contributions. So this is often the best balance for ctDNA-focused workflows. On the right is overspun plasma. Very high speeds applied indiscriminately can actually damage vesicles and protein complexes. Instead of cleaning the sample, you start losing or distorting EVs and altering proteomic profiles. So more spinning is not always better. Different analytes have different tolerances to centrifugal force. So centrifugation is not just a cleanup step. It defines the cell, vesicle, and molecular composition of plasma. Underspinning and overspinning introduce different artifacts, and inconsistent spin parameters across sites drive real cross-site biological variability. And this is where the table at the bottom becomes practical. It links centrifugation strategy directly to the primary analyte. For ctDNA, double-spun plasma is typically recommended because it minimizes leukocyte and platelet DNA contamination. For cell-free RNA or microRNA, you want gentler handling and careful pellet avoidance because RNA is fragile and cellular contamination can dominate the signal. For EVs, controlled low-speed spins help limit platelet-derived EV artifacts without destroying vesicles. And for proteins, moderate spins while avoiding extreme forces help preserve protein complexes and post-translational modifications. So centrifugation isn't one size fits all. The spin protocol has to match the biology of the analyte you care about. Even with the right tube and the right spin, biology continues to change while the sample waits, which brings us to processing time and temperature. This slide shows why delay is not just a logistics issue, it is an active biological variable. Starting with panel A on the left, that is looking at the delay before processing in the fraction of wild-type DNA. As time increases from immediate processing to hours later, you can see a steady rise in wild-type DNA signal. That rise is not tumor biology changing in the tube. It reflects leukocyte disruption over time, releasing genomic DNA into the plasma. The practical consequence is dilution of the tumor fraction. So if you are measuring ctDNA at very low levels, processing delay directly reduces your ability to detect it. Panel B shows the impact of delay on plasma microRNA profiles. This heat map is important because it shows that relative expression patterns shift early, even before total RNA degradation becomes obvious. That means holding time does not just reduce signal, it reshapes the profile. So two samples from the same patient can look biologically different simply because one sat longer before processing. Panel C moves to extracellular vesicles. These graphs show that handling conditions influence platelet-derived EV markers. As time and handling stress increase, you see higher EV signals consistent with platelet activation and residual cell release. This is ex vivo vesicle generation, not necessarily disease biology, so delays can artificially inflate EV measurements. Panel D focuses on proteins and hemolysis. These plots show widespread changes in measured protein abundance when hemolysis occurs. Red blood cell rupture releases intracellular proteins into the plasma, and that shifts many proteomic readouts. So even mild hemolysis during handling can look like systemic biological change. Across all four panels, the pattern is the same. Delay in handling do not just reduce quality, they actively change composition, and temperature controls the speed of these processes. Room temperature allows ongoing cellular activity and enzyme function. Cooler temperatures slow some reactions, but can also stress cells and alter release patterns. Stabilization tubes extend processing windows, but they do that by chemically modifying the environment. They do not perfectly preserve the original biology. They trade one set of changes for a more controlled, slower set. So the core message here is that time and temperature must be treated as biological variables. If they are not defined and enforced, the sample you analyze may no longer reflect the patient's in vivo state. Up to this point, we've been talking about how pre-analytical variables shape the biology of the sample. This slide shifts from mechanisms to strategy. It highlights a core reality in liquid biopsy. Single analyte and multi-omic workflows are fundamentally different design problems. On the left is a single analyte optimization approach. Here, every pre-analytical decision, tube chemistry, processing time, and centrifugation is optimized specifically to maximize signal to noise for one analyte. That will end up delivering the highest analytical sensitivity. It also simplifies validation and regulatory alignment, which is why this model dominates MRD assays and late-phase biomarker validation. The trade-off is biological scope. If you later want to look at RNA, vesicles, or proteins, the sample may not support it well without additional collections. On the right is a multi-omic workflow approach. Now the goal is broader biological context from the same patient sample. But this introduces compounding error risk because no single protocol preserves DNA, RNA, vesicles, and proteins equally well. Trade-offs across analytes accumulate. There are really two ways teams try to manage this. Strategy A is parallel collection, using analyte-specific tubes side by side. This gives the highest biological fidelity for each analyte, but increases cost and operational complexity. More tubes, more processing steps, more logistics. Strategy B is a single tube compromise. This simplifies collection for patients and sites, but it means at least one analyte class is being measured under suboptimal conditions. That is not automatically wrong, but it requires careful validation to understand what is being sacrificed. So the central point of this slide is that multi-omics is not just more data, it is a different workflow philosophy. You move from optimization to trade-off management. And importantly, this isn't just theoretical. The next slide shows published data demonstrating how different analytes respond differently to the same pre-analytical conditions, which is exactly why these trade-offs exist. This slide is here to make one point very clear. The trade-offs we just discussed are not theoretical, they are repeatedly demonstrated in the literature. The table is showing that there is a pattern and that is what matters. For ctDNA, white blood cell lysis increases background genomic DNA, diluting tumor signal over time. Even in specialized cfDNA preservation tubes, temperature and handling still change fragment profiles, so those tubes are not universally protective. For extracellular RNA, large consortium studies show that no tube reliably stabilizes xRNA across time points. RNA continues to be highly sensitive to handling. For EVs, platelet contamination can dominate the signal, and standard centrifugation protocols show huge variation in platelet carryover. Serum introduces even more artifacts through clot-induced vesicle release. For proteomics, processing delays lead to leakage of intracellular proteins, shifting protein signatures in ways that look biological but are handling driven. And hemolysis acts as a cross-omic confounder, altering proteins, metabolites, and nucleic acids simultaneously. So across analytes, the same handling conditions cause different failure modes. That is a key theme. Each analyte breaks in its own way under the same protocol. That is why parallel tubes or analyte-specific validation are not optional in serious multi-omic studies. So with that evidence in mind, the next question becomes how we design workflows that are actually trial ready. After everything we discussed about how pre-analytical variables can distort biology, this slide focuses on the solution, structured QC gates. The key idea is that pre-analytical QC protects biology, not just assay performance. It allows us to detect handling-driven problems early before they turn into false negatives or misleading biological conclusions. Think of QC in four stages. First is collection and handling. Documenting tube type, time to processing, and temperature conditions may sound administrative, but missing metadata can make it impossible to interpret unexpected results later. This is foundational. Second is processing and centrifugation. Early detection of issues like excessive delay or temperature excursions helps flag samples that may already be drifting biologically. Third is sample integrity. This is where we start measuring the material itself. Is the DNA fragment size consistent with circulating DNA? Is RNA intact? Are vesicle counts within expected ranges? These metrics tell us whether the sample still reflects biology or has shifted due to handling. Fourth is analyte-specific readiness. Before committing to final analysis, we confirm that the material is actually fit for the assay. The cell-free DNA yield is sufficient, RNA integrity is acceptable, or EV particle counts meet thresholds. Now the table brings this together in a practical way. It shows that QC requirements differ by analyte. Collection metadata and hemolysis checks matter across all analytes. Time to processing is especially critical for RNA. Fragment size metrics are central for cell-free DNA. RNA integrity measures like DV200 are essential for transcriptomics. EV particle counts and markers are specific to vesicle workflows, and proteomics requires its own stability and control checks. So QC is not one universal checklist. It is analyte aligned. And when applied early, these gates prevent both false negatives, where signal was lost, and false biology, where artifacts are mistaken for disease. With QC in place, the next step is design workflows that are truly trial ready from the start. This slide pulls everything together in a practical blueprint for moving from good science to trial ready execution. The first step is to define biological intent. Are we prioritizing MRD detection, mutation profiling, expression biology, or proteomic biomarkers? That decision drives every downstream choice. Second is selecting the collection strategy. That means deciding upfront whether we're using single analyte tubes, parallel tubes for multi-omics, or a validated single tube compromise. These choices must be justified and documented before enrollment starts. Third is standardizing processing. Time to processing limits, centrifugation parameters, and clear plasma definitions like PPP versus platelet-free plasma have to be locked and enforced across sites. Fourth, we apply pre-analytical QC gates. That includes metadata completeness, hemolysis checks, and free cell tracking, fail-fast controls that stop compromised samples before they enter analysis. Fifth is confirming analyte readiness. We verify that cfDNA yield and fragment size are appropriate, RNA integrity metrics are acceptable, EV counts make sense, or proteomic controls are within range. This ensures the material is fit for the assay. Only after all of that do we move to analysis and interpretation. Sequencing, proteomics, and multi-omic integration should only proceed when the biological foundation is defensible. At the bottom, you see the contrast between research-grade and trial-ready workflows. Research workflows tolerate variability and are exploratory. Trial-ready workflows are locked, enforced, and QC-gated. Trial readiness is designed at the start, not rescued downstream. When you look across these steps collectively, a consistent pattern emerges. Every upstream decision from tube selection through QC gating ultimately determines whether the final data reflects true biology or pre-analytical artifact. The implication is that success in liquid biopsy is not driven by any single step, but a set of disciplined principles that govern how the entire workflow is designed and controlled. I'll close with core messages to carry forward. First, pre-analytics determine sensitivity, specificity, and interpretation. By the time we sequence or run proteomics, much of the biological truth has already been shaped by how the sample is collected and handled. Second, there is no single workflow that preserves DNA, RNA, EVs, and proteins equally well. Multi-omic success requires prioritization and validation, not default compromise. Number three, centrifugation defines plasma. Spin parameters are not technical details. They define the cellular and vesicle composition of what you analyze. Number four, Time and temperature are experimental variables. They actively influence degradation, release, and stability and must be treated as controlled conditions, not logistics. And finally, QC before analysis protects biological conclusions. Early QC gates prevent false negatives and prevent us from mistaking handling artifacts for disease biology. If these principles are designed into the workflow from the start, liquid biopsy becomes trial ready by design, not by rescue after problems appear. So the key message to leave with is this. Trial-ready liquid biopsy starts long before sequencing. Pre-analytical rigor must be designed into collection, site execution, and specimen annotation from the very beginning. So with that, I'd like to thank everyone for your time and attention. What I hope you take away from this discussion is that liquid biopsy success is not just about the assay. It's about preserving biology from the very first step. The choices we make around collection, processing, and quality control ultimately define whether our data reflects true biology or pre-analytical artifact. There's a real opportunity right now in the field to move beyond variability and towards standardized, trial-ready approaches that support both scientific discovery and clinical translation. If any of these concepts resonate with the work you're doing, whether it's around multi-omic study design, pre-analytical strategy, or building more robust biomarker workflows, I'd be very happy to continue the conversation. Thank you again.